Thank you very much for inviting me in this exciting meeting. I am part of Spoke 8 of Tuscany Health Ecosystem, and I will try to show you the results that we obtained up until now. After the very nice introduction from my colleague, I would say that in the human genome we have around 2,000 genes, which means we can have 2,000 or more proteins, considering also the alternative splicing. And we expect quite a large number of potential drug targets, considering both proteins, nucleic acids, and so on and so forth. However, unexpectedly, the number of drug targets of all approved drugs is quite low. In this analysis of 2006, you may see that 324 drug targets were known, which account both for the human genes and the genes of our pathogens. In the 18 years that passed, the situation is not dramatically changed because we have around 400 drug targets. This means that we may have quite a lot of space for innovative drugs. We should also consider that most of the drug targets that are used nowadays are enzymes. Around 50% of them are enzymes, around 30% are G protein kappa receptors, 7% ion channels, and so on and so forth. Enzymes, well, I make this very small introduction for people who eventually are not active in drug design. Enzymes, we know they are biological catalysts. They are involved in most metabolic and physiological processes. Imbalances in their activities, either up or down regulation, cause diseases. And of course, we can act with inhibitors or activators of enzymes to correct such defects. And there are many examples of widely used drugs which are, in fact, enzyme inhibitors, like the painkillers like aspirin, ibuprofen, indomethacin, and so on and so forth. Most anti-infectives, antibiotics, antivirals, antifungals also belong to the enzyme inhibitors. Agents for the treatment of high blood pressure, phosphodiesterase 5, and I could continue with very many other examples, but also many anti-cancer drugs belong to this. So we are involved in the neurodegenerative diseases only in the last several years. We know that there are different diseases which are characterized by protein misfolding. The most relevant one is Alzheimer, on which I will speak, but it's not the only one. There are also diseases like Huntington's, Parkinson's, the prion disease, and so on and so forth. Usually there is an issue with the aggregation of proteins. Normally the misfolded or badly folded, unfolded proteins are recognized and they are degraded at the proteasome level, but there are situations in which they accumulate, and this leads to problems, to health problems. And the most investigated one is, of course, Alzheimer's disease. It's a disease with few therapeutic options nowadays. Even if some monoclonal antibodies have been approved recently, their efficacy is rather limited. We know that it's an irreversible progressive brain disease which destroys memory and thinking skills. And what it's typical for this disease is these structures. These two structures that I show here, one it's called beta amyloid plaques. They are formed outside the cell and they contain a protein called amyloid beta with other materials, cell material debris, and similar things which accumulate around the cells. Whereas the second structure, it's called neurofibrillary tangles. They are formed inside the neurons. They are twisted fibers that build up inside these nerve cells, and both of them interfere with the normal function of the brain. Alzheimer's disease is characterized also by other abnormalities, for example, in the neurotransmitter system. The most affected one is the cholinergic and glutamatergic transmission, but also there are some reduction in the monoamine neurotransmitters, that of some peptides, whereas the inhibitory, the GABA neurotransmission, is not much affected in these conditions. Excuse me. And obviously, being such a terrible disease with few therapeutic options, there are many drug targets which have been proposed over the years. They include some enzymes like the cholinesterases, the monoamine oxidases, some neurotransmitter receptors, and the mitochondrial damage, and so on and so forth. You see quite a lot of them, but none of them, unfortunately, works well for the moment. A few years ago, in collaboration with colleagues from my department and an Italian-American scientist, she's originally from Florence, but she's active at Temple University in USA, we proposed carbonic anhydrases as drug targets for Alzheimer's, and I will show you why we have made this proposal. So there are clinical data which show that carbonic anhydrase is downregulated in the brain of many patients. So there are data in post-mortem patients for which the levels were measured. The enzyme which is present was also shown to be nitrated or oxidized. This leads to impairment of its functioning. And also the plasma levels in some patients still living with this disease were showing that there is a decreased, I think there is some noise probably from outside. Anyhow, so this is one of the reasons why we investigated carbonic anhydrase in Alzheimer's disease. The second issue is that in the last 10 years, we show that carbonic anhydrase is involved in modulation of memory. There are implications for the treatment of many cognitive disorders. So there is work which we have done with the group of Patrizio Blandina, Gustavo Provenzi, in which we show that the carbonic anhydrase activation is important for the recognition memory, of course in animal models, for the extinction of fear memory, and so on and so forth. So there are strong connections. But what is carbonic anhydrase? Well, these are enzymes on which we worked for quite some years. They catalyzed a very simple reaction, the hydration of carbon dioxide with formation of bicarbonate and protons. It's a real simple reaction. You may ask, why do we need that? Because it's very slow at physiological pH. The enzyme, it's catalyzing efficiently. We have 7 orders of magnitude an increase in the formation of bicarbonate and protons in the presence of the enzyme, which explains why there are 8 genetically distinct families encoding for them in organisms all over the phylogenetic tree. Clearly, from 2 neutral molecules, you obtain a weak base, bicarbonate, and a strong acid, the protons, and this means that the enzyme is mainly involved in pH regulation, but not only, it's also involved in metabolism. We know in detail the catalytic mechanism of all these enzymes, so we work with representative of all 8 genetic families. We know the active site architectures, but I will not enter into too many details. In humans, there are only alpha carbonic anhydrases of the 8 genetic families I mentioned, but there are actually 15 different isoforms, which is nice from the point of view of investigating them, but it's also complicated for medicinal chemists because we have to design inhibitors and activators which are selective for them. You may see that they have different localization within the cell with cytosolic, mitochondrial, membrane-bound, and secreted enzymes. They have different activities, and they are involved in different diseases, but I will not enter into too many details here. So in the brain, we have 11 of the 14 isoforms, and we don't know why we have so many isoforms. We hope to determine this with the help of this project in which we are involved. And clearly in the central nervous system, they are involved in pH control of the brain, which is a tightly controlled process as in all organs, in excitability of the neurons, in the production of cerebrospinal fluid, and also in the regulation of cerebral blood flow. In fact, inhibitors of these enzymes are in clinical use for decades as antiepileptics for the management of idiopathic intracranial hypertension and for other neurological conditions. But what we are interested in are the activators, because as I mentioned, they are involved in cognition. And in fact, in the late 90s, it was shown by a colleague from Finland that in the presence of the activation, the GABAergic transmission, the GABA, we know very well, is the inhibitory neurotransmitter, becomes excitatory, which may explain why we have an enhancement of the cognition in the presence of the activation of carbonic anhydrase. Of course, the mechanisms are a bit more complicated, but just to simplify them, I show that. And the pharmacology of the activators is also rather complicated. I will not mention everything, but in 2017, with the group of Patrizio Blandina, we showed that the activators which were administered to an animal model helped an enhancement of the object recognition memory. And we also found how this occurs through the activation of a kinase, more precisely the ERK kinase, in the cortex and hippocampus of these animals. Later, we also showed with the same group and the group from Brazil that the activators are enhancing the consolidation of the fear extinction memory, which may lead to interesting opportunities for the pharmacological treatment of diseases for which there is not an appropriate treatment. In the project in which we are involved in Spoke 8 of THE, we proposed a design strategy of modulators of the enzyme activity within the brain, using both activators and inhibitors of the carbonic anhydrases, which were hybridized with different other chemotypes, for example, cholinesterase inhibitors, monoamine oxidase inhibitors, and so on and so forth. So of course, the chemistry is nice, but I will not enter into details. Why? Because we tried to hit two different targets with just one molecule. And here are some examples of the different series of inhibitors and activators which we have obtained. There are my colleagues, some of them who are here, Paola Gratteri, Alessandro Centino, Alessandro Bonardi, Andrea Amara, who are involved in the project. You may see the different series which include monoamine oxidase B inhibitors, hybridized with a carbonic anhydrase inhibitor, cholinesterase inhibitor, carbonic anhydrase activator, and different other combination of these possibilities. All of them include series of 40, 50 different compounds, which were, of course, investigated and evaluated in different models. I will show you only two recent examples of, let's say, rather successful ones. The first one involves the hybridization of monoamine oxidase inhibitor of the coumarin type with a carbonic anhydrase inhibitor of the aryl sulfonamide type. With colleagues from our university, Gustavo Provenzi and others, we showed in the pharmacological models that they are helpful for protect against amyloid beta-induced neuronal toxicity, which is obviously a model of Alzheimer's disease. They also reduce oxidative stress and mitochondrial dysfunction, which are typical of this disease. I will not enter into too many details, but the references here for who is interested. The second example, it's again a recently published paper just a few weeks ago, in which we observed enhanced recognition memory through the dual modulation of brain carbonic anhydrases with activators. You see the fragment contains, excuse me, the fragment contains carbonic anhydrase activators. I made an error. No. Okay. So we have inhibitors of the cholinesterases of the tacrin type and activators of carbonic anhydrase of different types. And also in this case, we have nice results in an animal model of cognition. Obviously, the compounds have been tested as activators of several brain-associated carbonic anhydrases of cholinesterase and butyryl cholinesterases, and we have nice expected results of the activation and inhibition. I was mentioning the pharmacological model, again done by Gustavo Provenzi. This is a social recognition model in mice, and again we have pro-cognitive significant effects when the mice were treated with these compounds, which were better than the combination of the two different drugs or of the drugs taken separately, obviously. I wish to thank my collaborators and the financing from this project, and thank you very much for your attention.